lecture I like to call The Good, the Bad, and the Ugly for B440. These slides, information can be found in Chapter 11 on host microbial interactions, also in Chapter 1 on the microbiota or the human microbiome, and the rest of the information is from primary research articles, some of which I've referenced in here. Others, you can ask for the references if you want to read them. In this lecture, in this set of slides, we will be talking about the good, the bad, and the ugly, and the hygiene hypothesis. The good guys are the guys that are normal flora, or the residents of the human body. This picture here shows a couple of them that are really commonly found in probiotics, including Lactobacillus acidophilus and Bifidobacterium longum, the good guys. The bad guys I consider dysbiosis. That means an unequal balance of the things we call pathobionts, which I'll talk about in a little bit, and the normal flora. And then the ugly, which are the actual pathogens. In this picture, down at the bottom would be the ugly ones. Mycobacterium tuberculosis, which obviously causes tuberculosis, Clostridium tetani, and you can guess that would cause tetanus. Yep. These are two examples of pathogens that can invade and infect a human, but are not commonly part of the resident biota. And then we'll get into hygiene hypothesis in a little bit. So let's start with human microbe interactions. And I'm not going to do what I will do with you in a face-to-face lecture, but just consider how complex the interactions are between every organism on our planet. That would be us and plants and animals, bacteria, viruses, fungi, right? We are constantly in interaction with everything in our environment, but especially with microorganisms. That's in the external or exogenously sourced, the environmental organisms. And these are not just bad guys, these are not just pathogens. These are good guys too, or stuff that doesn't cause any damage, any harm to us. If you're breathing right now, you are breathing in hundreds of thousands of different spores or prokaryotic cells, virus particles that are both animal viruses and bacteria viruses and plant viruses. There's tons of stuff in the air, in the foods we eat, in the water that we drink, in the bed that we lie in at night. Those are exogenously sourced organisms, things that are from the outside environment and can interact with us on the outside or even invade on the inside. But also we have an endogenous biota. And endo means inside. Genus is originating in. So originating in the inside, which means resident biota, the organisms that we are colonized with early on and that stay with us for our lives. And these resident organisms can flux and change and do so on a daily basis, even on an hourly basis with our diet, if you're talking about gut bacteria or with our respiratory tract. But these are endogenous biota and organisms that normally are found in a particular balance with the host, which is us, our immune system, but sometimes can shift and cause an imbalance, which will lead to some pretty nasty things, which we'll talk about extensively throughout the entire semester, but also some in these lecture slides. This last part, the body hosts a wide range of microbes because of diverse habitats. So what does this actually mean, diverse habitats? How does the human body have diverse habitats? Well, I mean, if you think of an ecosystem, what defines the habitats within any ecosystem? And if you are in that face-to-face lecture, we'll talk about this quite extensively, but just thinking about it, right, you would describe an environment or a habitat based on things like temperature or salinity, how much salt is in the air or in the water availability, which would be like moisture, right? All of these things that would describe like an ecosystem outside the body can also be used to describe different parts of the human body on the inside and even on the surface, right? So for example, temperature. The temperature of our airways is much different than the temperature of our deep bowels, like our colon. Colon is one of the warmest regions of the body, and the airways is actually dependent upon environmental exposure. salinity, right? How much salt there is. That's different whether you're talking about the surface of the skin, which is highly salinic, so a lot of salt concentration there, or like saliva, or somewhere inside our gastrointestinal system where we don't have a high concentration of salt. So these particular abiotic factors can really define a habitat, and therein kind of define what types of organisms will reside in each of those areas. And this is kind of why, actually, this in large part explains why, if you have a cold, why if you spit into your open wound on your ankle, you won't get a cold on your ankle. It's because mostly that virus has a very particular preference to the upper respiratory tract. And in that case, the virus is very particular to host cell receptors. And those receptors are just not found on the same cells that are in your ankle or in your blood as they are in your throat. But also because viruses and bacteria are very sensitive to their ambient environment. So they really do require a very particular range of temperature, pH, etc. So let's start with the good guys, normal flora. These are colonized bacteria adapted to the body. And this picture kind of gives you an idea of what we're talking about. There's a lot of emerging research talking about the benefits of normal flora, and that's in large part what these slides are going to be talking about here. A great segue for you to do is to think about other names for normal flora. This is not just a good practice for you, but also very important in really setting yourself up for the rest of the semester in this course. We will be talking about the normal flora extensively throughout pretty much every chapter, and we'll use a lot of different names to refer to these guys, like resident biota, as I've already used, or the microbiota. Microbiome is a little bit different. It refers to the set of genetic information inside the human body. But hopefully you can work on this question on your own time. So where in or on the human body do we find the normal flora? This table is from at least one version of the textbook. I think this is from the current edition, from chapter 11, Shifting Understanding of Sites with Microbiota. So the first part of this table talks about sites that are known to harbor normal microbiota. And these are areas that we have been able to essentially isolate out and grow organisms to determine if there are viable, like living bacteria in each of these areas. Now, the other two sections, additional sites now thought to harbor at least some normal biota or their DNA, and then at the bottom, sites in which DNA for microbiota has been detected. Now, these two are a little bit more questionable as to whether there is an actual resident flora in these different areas. In fact, if you look at the research, the current research to date still is not quite sure if we can identify, for example, a breast milk microbiota, right? If there are actually microbes that live regularly inside that milk environment. That's actually one of the key areas of my research, so I can talk a little bit more knowledgeably about that. And I will say that yes, indeed, we can isolate out organisms from each of these areas, but whether or not they're true endogenous flora, meaning they're adapted to that environment, they exist there readily, and they have some type of functionality, is still not quite understood, not known, not really even agreed upon. But that first section, really anything that's exposed to the environment, right, outside and inside, is going to have a set of organisms that are adapted to that particular area. Skin and adjacent mucous membranes is a huge area, and we'll talk about that a lot more actually in our immunology section. Upper respiratory tract, that's like your throat, your kind of your sinuses too. These do have some organisms that exist in that area. Gastrointestinal tract, really from start all the way through the end. In fact, your gastrointestinal tract has the highest percentage of our normal flora. That's where we have the most abundant numbers of organisms, specifically in our colon, in our large intestine. The urethra, external genitalia, vagina, external ear canal, and external eye. What would we call it if you had organisms in the internal ear canal? Yeah, exactly. This is otitis media or an ear infection, right? So it is important to understand where you would have normal flora versus if you find a bacteria that are not resident to that area, it would be an infection. So when does colonization begin? the current and still mostly accepted understanding is that in utero, the fetus is sterile, so there are no maternal microbes that are getting into the baby until those membranes break. And as soon as the membrane breaks, then you have birth canal exposure immediately, and that's regardless of whether the baby is delivered vaginally or through cesarean section. As soon as those membranes break, that is the first and initial exposure. And you do find a significant difference actually in babies that are born after the membrane breaks or before the membrane breaks through surgical delivery, through cesarean section. And as long as that membrane has broken, then there is a significantly altered microbial colonization. So that's within that very, very initial exposure period. Once that baby comes out, there's environmental exposure period, again, regardless of mode of delivery. So that would be the manhandlers, the people in the hospital, if the baby was born there, or in the home if the baby was born there. The actual environment, the surfaces that the baby is put on, the things that the baby touches, all of those things have very low levels usually, sometimes high, depending upon where the baby is in the world, levels of microbes. And you'll find many of those microbes transferred into that baby right away. And so within the first 8 to 12 hours, the baby is initially colonized with about 10 to the 9th bacteria. And if you're trying to figure out what that number is, that means how many bacteria are essentially in 1 gram of feces, if you're thinking about it in that way. So it's quite a bit, but it's still actually quite a low number compared to what our ultimate colonization is. The little asterisk mark we have here, sterile until membrane breaks, right here. I put this here because there still is some question as to whether or not the baby is truly sterile in utero. So here, placental microbiome and its role in preterm birth. Now, these articles are actually a little bit old, but there still is emerging evidence that there might be some environmental exposure even inside the womb. Is meconium from healthy newborns actually sterile? If you don't know what meconium is, this would be the first bowel movement essentially of the baby, and one that physicians use to really try to figure out what the uterine environment looks like, because the first bowel movement comes usually from stuff that the baby had been exposed to inside the womb. So for a long time, meconium was believed to be sterile, but actually for about 10 years now, we know now that there are low levels of organisms that can be detected, at least with the DNA level. Whether or not these are culturable, like live bacteria, is still sort of in question, and we could talk about that in more detail at another time if you'd like. But these are just evidence that there still is some question as to when we get our first exposure. But as mentioned, it's still a generally accepted thought that the baby is sterile in the womb until birth canal exposure. This picture just gives a visual for what we just mentioned. So where are those sources of the initial seeding, essentially, if you want to think about it that way? Where does this baby get their gut bacteria or respiratory tract bacteria from after or during delivery? So mode of delivery is a big one, and babies who are born cesarean section versus vaginally delivered do have a different set of microbiota, at least in that first week of life. As mentioned, if the baby is born through surgical delivery before the membranes break, so voluntary C-section, they also have a completely different set of organisms as to babies who are delivered essentially through emergency C-section, who have already been exposed to that birth canal. The caregivers, so the other parents that are around that baby, siblings, aunts, uncles, friends even, do have a significant contribution to the microbiota inside that baby. Caregivers, or I guess healthcare workers, of course, right at the time of delivery, but also even that first month of life can contribute to that microbiota. And then here, diet, which is what we're looking at with both of these pictures, so formula versus breast milk, has probably the most significant contribution to early life microorganisms inside that baby. So let's talk about some of the benefits that normal flora can bring. And I do want to bring to your attention, although I don't have the slide on this particular set of slides, we do have key concepts for Bio440, and these key concepts are concepts that I have essentially deemed worthy for master level understanding that you will be tested on at every single exam during the entire semester. And this would be the first key concept. So what are some benefits of the normal flora? You should basically try to memorize, if you can, all of these key categories that we'll mention in the next few slides, and at least give some examples of each of these. So, for example, one category of the benefits of normal flora would be colonization resistance. And the concept here is the protection from pathogenic colonization. And so for you to come up with a couple of examples, I've given you 4 sort of subcategories with their own examples. So if you can remember a couple of these things, or even as we go through the semester, we'll talk about these in more detail and give other examples of these. So any way you can remember some examples of each of these different categories, that would be to your advantage for sure. You will be tested on it, as mentioned, at least 4 times throughout this semester. All right, so colonization resistance. Here are some ways that normal flora can protect from pathogenic colonization. And most of these things that we'll talk about here are based on the gut bacteria because, as mentioned, this is where the majority of the normal flora exist on the human body, and also where the majority of the immune system really sets up shop in. And then that would be your large intestine, some in your small intestine too, but really we're focused on the large intestine. So here in the large intestine, there are fermenting bacteria, so lactic acid bacteria, actually that's true in the small intestine too, can drop the intestinal pH down to even pH of 3. There's a really awesome example of this bacteria I'm going to mention in just a minute, Bifidobacterium infantis, for infants who are breastfed. If infants who are breastfed receive this as a probiotic, this bacteria can colonize their colon, and there's been some really cool studies on the infant pH, or fecal pH at least, where that pH has dropped even from like pH of 6 all the way down to pH of 3 just from the presence of this one bacteria. And this low pH is really beneficial to reduce pathogenic invasion, even infection, as these pathogens really have a hard time in that low pH. That second one physically covers attachment sites. That should be pretty self-explanatory, but really microbes are just like any other organisms on our planet. They need a place physical to set up shop, to bind to on the surface of our epithelium, whether that's your skin, your respiratory tract, your digestive tract, and from there to replicate and make more copies of themselves. So with an abundance of normal flora, there is very little attachment sites for pathogens. Now this of course can be compromised if you have a perturbation. For example, antibiotic treatment will wipe out a lot of your normal flora binding areas, and wipe out a lot of your normal flora, which will essentially just give wide berth for pathogenic colonization. This third one here, secrete antimicrobial chemicals. So an example of this is called microbial antagonism, where 2 free-living organisms are essentially competing for resources, but in this case, one is actually antagonizing another. A great example of this is Lactobacillus reuteri, which is a common small intestinal commensal bacteria, and this particular one secretes something called a reuterin, which is antagonistic towards a lot of gram-negative pathogens. And this last one here, outcompete for resources. An example of this would be that bacteria I mentioned already, Bifidobacterium infantis. This is a key or champion colonizer of the infant intestinal tract because they have the ability to consume all of these really unique components of milk called human milk oligosaccharides, which are essentially fibers that feed beneficial bacteria, and this prevents pathogens from accessing really almost any of the sugars that are available in milk. So these 4 kind of subcategories are examples of how normal flora can provide protection from pathogenic colonization, but this is not an exhaustive list. So we're talking about the benefits of normal flora, and in this image is just to capture that spirit of colonization resistance, where if normal flora are kept pretty happy inside the body and on the skin and the respiratory tract or in the gastrointestinal tract, it's very difficult for invading pathogens to set up shop. Second benefit of normal flora is improves metabolic activity or metabolism in the host, so it's in the human. So here are some examples of this: consuming indigestible fibers, producing usable byproducts. So many examples, or many bacteria are able to do this, but really commonly the family Clostridialis, There are many organisms within this that are able to do this, but not all are, many are beneficial for the human. So these guys can, inside the colon, they receive fermentable fibers, things like inulin, oligosaccharides like galacto-oligosaccharide or fructo-oligosaccharide are some examples of these types of fermentable fibers that get into the colon. Our body doesn't do anything with them because we don't have enzymes that can cleave off those sugar molecules from those long oligosaccharides, but bacteria do, and they can clip these sugars off and consume them, and they ferment them into these short-chain fatty acids, including butyrate. And this one in particular, butyrate, fuels colonocytes. It actually has a whole bunch of benefits, but this is one example of them. Great thing about this particular molecule, butyrate, can actually convert colonic cells from a metabolic process that produces oxygen, so making the colon kind of oxygen rich, to doing an anaerobic process of metabolic activity, so that they essentially are producing or creating an anoxic environment, which is really detrimental to most pathogens. There are very, very few pathogens who are able to grow in an anaerobic environment, a completely anaerobic environment. So these fermenting bacteria are not only beneficial in reducing the pH, but also in helping shift over our metabolic activity of our colon cells to an anaerobic process to generate that anaerobic environment. And also, this really promotes colonic cell turnover, which means it really boosts the metabolic activity in our colon cells to help regenerate our colon cells. So there's a lot of shedding of our cells at the surfaces of our colonic region of our large intestine, and this is beneficial because our colon is really the site of like where most of the toxins kind of aggregate together as they're built up into our fecal samples or to our feces to be removed from the body. So they're exposed to a lot of really nasty things, and they get damaged in the process. So it's very beneficial for us to have a rapid turnover of these cells so that any damaged cells get essentially removed and replaced with fresh new ones. The second one here helps regulate blood glucose levels. So there is evidence, and again, this sort of has to do with these indigestible fibers. So if you have fibers in your diet, these bacteria can really help process these fibers and have really a slower release of glucose that's broken off from some of these fibers, and reduces that spike, postprandial, which means after eating, that you would experience in blood glucose levels. And actually, if you look at any of these reports on, the microbiota in diabetics, you can find that changing the diet to a high fiber content, which promotes a beneficial bacteria in the gut, if you're diabetic, this alone can really help moderate the glucose intolerance and even provide some therapeutic impact in these individuals. This third one here improves gut transit of food, right? So if you have beneficial flora, and again, this all really has to do with diet. So as long as you're introducing dietary components that really promotes the growth of the beneficial flora, then that in turn results in benefits for you, right? So improvement of gut transit, which means the food doesn't get stopped up, so no constipation, and it also doesn't get pushed too quickly through, which would result in diarrhea, right? So one or the other, but it really helps moderate this. And this last one provides essential nutrients. A really classic example of this is E. coli, which is actually in really low abundance in the human body, but almost all of us have, actually I would say all of us, but I don't know that for a fact, but the majority of humans do have a very small amount of E. coli. This usually represents less than 1% of our normal flora, but still it's there. It's very well known that E. coli can produce vitamin K, which is essential for blood clotting factors. So it's one of our coenzymes necessary for thrombin and prothrombin, for example, in blood clotting. Folic acid synthesis, right? We humans do not have the ability to make folic acid, which kind of is part of our B vitamins. It's important in nucleotide synthesis, etc. But bacteria do. They can produce folic acid and then secrete it out, and we can use that folic acid. So these are a couple of examples of essential nutrients that bacteria actually produce for us. If you've ever thought about it, right, babies receive a vitamin K shot if they're born in the hospital before they are released to go home. We know this. And one of the main reasons is because these babies, if you recall from our conversation just a few minutes ago, babies don't have a very high number of flora, their endogenous flora, right? They have to get colonized. It takes 8 to 12 hours to even get a low number, 10 to the 9th, and then another 24 hours to get up to 10 to the 12th bacteria, which is kind of the normal level for the colon. So within that period of time, they actually have no or very low vitamin K synthesis because it comes from the bacteria in the gut. So we provide them a vitamin K shot just in case, right, there's any problems with a cut or a wound, so that they do have appropriate blood clotting within that short period of time that the bacteria inside their gut are not yet producing enough. It's kind of interesting. Here's a picture more to talk about the diversity of the diet. So if you're wondering how to gain all these benefits I'm talking about to get this diverse flora rather than a very simple monocolonized gut, simple diets are associated with simple numbers of bacteria. So monocolonization, or very, very low abundance, diversity, of bacteria, which provides, very low benefits. But to gain diversity and to gain the immense benefits that we're talking about, really your diet must be as diverse as the diversity you are trying to aim for in the gut flora, and to gain all those benefits. And especially looking at this type of diet that is depicted here: nuts, grains, fruits, veggies, legumes. I mean, you don't see huge dairy here and huge animal products here. It really just focused a lot on these fiber-rich foods. Third category of benefits of normal flora: improves immune function. And as with all of these slides, I'm just giving you a little snapshot of some of these benefits and ways that this really plays out. But each of these little categories we can spend months talking about with the literature that we have out there and what we know now about the benefits of these normal flora. So just keep that in mind as we talk through this. So some examples that we know of how the normal flora can improve immune function. This first one here: induces immune tolerance and regulation. These terms here, tolerance and regulation, they actually mean two different things, but both are really, really important in an appropriate immune response. Immune tolerance does sound exactly like what it is. So it helps train our immune system to tolerate certain types of things. We do this on a regular daily basis. If you eat food, food is foreign, and therefore our body's immune system recognizes food and all of those things that food has, all the proteins and lipids and etc., as foreign, and will mount an immune response against all of those food items if it is not trained to tolerate this. So we call this oral tolerance, and in fact our normal flora seem to be really, really important in helping promote immune tolerance. That's not just to food things, but also it is to the resident flora. Like if we don't have beneficial bacteria at an appropriate level and right in the appropriate sort of balance, then our immune system is not trained appropriately to recognize those bacteria as normal, and therefore can mount an immune response to even the beneficial guys. And then regulation here. Regulation really more means the ability to respond appropriately to pathogenic things and not inappropriately to things that are benign, like your own self stuff. That would be autoimmunity, or like food particles, right? That would be food allergies, or normal flora, right? So things like this. So it's really trying to regulate the response of the immune system so you have appropriate activation when needed, but down regulation or dampening of the immune response when it's not needed. So an example of this is this one really, really cool study that really showed one particular species, Clostridium ascendens. This guy produces propionate, which is another type of short-chain fatty acid produced in response to fermenting fibers, some very particular ones. And this propionate induces T regulatory cell development. So T regulatory cells, we'll talk about more when we get into our chapter 13 on the adaptive immune system. But these ones, again, they are what they sound like. So they are really, really important in helping regulate our immune system. So these T regulatory cells actively secrete out chemicals that dampen our effector cells, the ones that actually go out and do stuff like, cause an immune response. So the fact that this particular short-chain fatty acid produced in response to fermenting fibers from this particular organism really promotes the regulation of our immune system is quite fascinating. In fact, we can find this in these mice called gnotobiotic mice, or mice that are germ-free, so mice born in a germ-free facility, and so they've never been exposed to pathogens. They've never been exposed to microbes at all, and they are raised in germ-free facilities, so they never have any normal flora inside any part of their body. And what you'll find is that these mice, they really don't succumb, of course, to pathogenic infection, but what they do experience is a huge increase in, percentage-wise at least, autoimmune disorders, right, cancers, these really strange things that you don't normally see in mice develop because they don't have this factor here. They don't have the normal flora inducing tolerance and promoting regulation of the immune system. So the immune system really just goes out of whack in these mice that don't have normal flora. There's a lot of really cool studies on this in so many different ways, so trying to deduce when this is most important, and it does seem to be really at early age, so like really even during pregnancy, but right after birth, when you have that initial flora training the immune system. Same thing here, trains the immune system to not react to benign antigens. And this word benign, if you're not familiar, it just means like non-toxic or non-pathogenic, right? Things that don't cause damage. And this last one reduces allergies, autoimmunity, upper respiratory tract infections. I mean, all of this stuff is very much related. It just really helps promote a regulated immune system. And you'll find that even like beneficial gut bacteria help reduce upper respiratory tract infections overall, which is quite fascinating. But it really drives this concept that the majority of our immune system stems from our gut, in the same place that the majority of our microorganisms are. So what's happening with our normal flora in our gut really does have a significant impact on whole body function. Everything in our body, our cognitive, our brain, our immune system that goes from top to, the top of our heads down to the bottoms of our feet. All of this stuff has a ton to do with what's happening in your colon. And this last category is brain function. As just mentioned, cognitive development is significantly impacted by what's happening in your gut. So the importance of your diet in not just balancing your mood and behavior through foods, but specifically through the molecules that your gut bacteria are secreting. We'll have a presentation towards the second half of the semester that's on the gut-brain axis by someone in this class, and we'll hear a lot more details about many of the different ways that the gut bacteria can impact the brain, just as we see in this picture. And of course, we're not just talking about the bacteria here. In this picture, it really does depict a lot of different types of organisms can have a significant impact on cognitive development, mood and behavior, or brain, just brain function altogether. And we call this the gut-brain microbiome axis. You may have heard this from, actually for decades, this has been labeled as the gut-brain axis, but because we know now that the microbiota, especially of the gut, plays such a significant role in brain activity, we now are discussing this axis as having this third arm. The gut-brain microbiome axis regulates hormone secretion, even improves satiety, and regulates weight in this manner. So there are chemicals that are secreted during the process of digestion that are involved in satiety. This is why we don't keep eating, most of us. When we've eaten a certain amount of food, like CCK is secreted in our gut, and that is a satiety hormone, so it tells our brain, like, stop eating, we're not hungry anymore. Gut bacteria are known to, or I would say very particular groups of gut bacteria are known to help induce these hormones that improve that feeling of satiety. So even before your body would do it naturally, the gut bacteria can help promote that. Benefits of normal flora and sort of what defines normal flora. Now let's go all the way to the other end of the spectrum and talk about the ugly, right? The good, the bad, and the ugly. So the ugly, the really bad guys, are what I consider the pathogens. And pathogens, and we'll pull this, I'm pulling this out of chapter 11, which will go into a lot more detail in the next set of slides. But pathogens are defined by these two major factors. Number one, the ability to establish in a host. And this is not as easy as it sounds, right? These are all exogenously sourced. That means they come from the outside of the body. If we're talking about, like in this example that I have here, these three different types of pathogens, these are enteropathogens, which means pathogens of the gastrointestinal tract. You'll hear me talk a lot about these things, in part because most of our flora, as mentioned 20 times already, exist in the colon or the small intestine, so the gastrointestinal tract, which is where we find a huge amount of pathogenic bacteria, right? They get into our body through our oral cavity, and they can set up shop in the gut. But also because this is what I study, so I know a little bit about these guys. So let's just talk about gastrointestinal pathogens like these three here. This is Salmonella, Shigella, and EHEC, or E. coli, enterohemorrhagic E. coli. So how do they get in? Well, first they have to pass, of course, through the oral cavity, go down the esophagus, through the stomach, through the small intestine, and somehow end up in the colon. And what are the things they have to pass through? Well, they have to get in, right? So somehow they have to make it from the colon of an infected individual and get into the mouth of a susceptible host, right? That would be the fecal-oral route of infection, which is as disgusting as it sounds. So they have to get in. They have to migrate through all of our defense systems. So in our mouth, we have saliva, which actually does have antimicrobial molecules in it, like lysozyme, that kills, that just degrades the peptidoglycan, the cell wall of bacteria. If they can pass through our oral cavity and even the mechanical digestive capacities of our tongue and our teeth, they pass into the most toxic environment imaginable, which is our stomach. So our stomach has tons of digestive enzymes in it, but of course the most significant component is that low pH, so pH of down to pH of 2, which is extremely acidic and very, very harmful for almost every single organism. And most pathogens die here. They just cannot tolerate. But if there's a high enough number of pathogens, so maybe you took a bite of a burger that was just loaded with salmonella, not salmonella, like something like Shigella or EHEC, then you just have a huge number, and not all of them would die right away, right? Because of the time it takes to transit that food through the stomach into the small intestine. So perhaps a contingent of them survived into the small intestine. But here again is an onslaught of bile acids and more digestive enzymes that really just emulsify most things going through, right? And if they manage to pass through all of that, they have that colonization resistance factor of the normal flora, not just the difficulty in establishing, like finding a binding site, but protecting themselves from innate immune components and adaptive immune responses from the human and antimicrobial molecules from the normal flora. And if they're able to bypass all of those defenses and find a spot, then they're able to establish in the host, right? They have to physically attach to, bind, and then begin to replicate. So that's all encompassed in number 1, the ability to establish in the host. And once they get there, they're really not considered pathogens until number 2, the ability to cause damage. So that would be toxins that they secrete, right? Those are exotoxins or even endotoxins, part of their cell component, like lipopolysaccharide, which all 3 of these bacteria have. Most pathogens have some type of toxin, something that they secrete out that's damaging to the host cell or the host environment. And we'll talk about those in a little bit more detail. These in particular are images of 3 different bacteria. This is Salmonella, this is E. coli, this is Shigella. Now the cool thing is all of these guys are exemplifying a really amazing factor in this ability to cause damage. So E. coli, for example, forms these pedestals, and this is actually this is our epithelial lining, so all 3 of these are our colon or small intestine. And all 3 of these guys have the ability to cause changes in our cytoskeletal arrangement. Cytoskeleton is what really defines the shape of the cell and the movement of the cell. And so these guys really, they secrete out some type of chemical, all 3 of them do. And they're essentially just saying, hey cell, I'm ready to bind to you. And this cell changes and creates these really amazing formations. So in this picture here, this is called a pedestal formation. So they cause the cell to create these little pedestals that they just rest upon, and they replicate. So this is their binding, and then they begin their replication. For these guys, you can see these are the microvilli, the small intestine, and they're significantly altered with this bacteria, the bacteria in the pink. So there's a really major change that these Shigella cause for these small intestinal cells, and a huge disruption in microvilli. And then for Salmonella, they cause this ruffling. So they knock on the door of the epithelia, secrete out these chemicals that cause the cell to just sort of open up its face so the bacteria can just climb right in. So all 3 of these are examples of damage caused to the either colon cells or small intestinal cells, which causes a significant amount of damage. So let's talk about germ theory for a minute, and you'll find this in chapter 1. So the concept here is that one pathogen causes one disease. The history of this, you can read about it, goes back to the 1860s with Louis Pasteur and other scientists who really began to pinpoint diseases and disease-causing tiny agents, right? And there's some really, really cool history behind this, and I urge you to go read about it if you have the time to do it. And this has been pivotal in really helping us control infectious disease in human society. This is why we don't all die of influenza, because now we have an understanding. It's not that we've adapted necessarily to the virus, but we have an understanding of what causes these infections and how to identify and how to prevent these infections. Right? So this is pivotal to modern or to classic medicine, traditional medicine. One pathogen causes one disease. Those three examples I just gave: Salmonella, Shigella, EHEC. All of them can be identified as causing a very particular type of gastroenteritis, a particular type of intestinal disease. And they're defined by particular signs and symptoms that help diagnose that causative agent. So all of that stuff is important. But today, the problem is that this does not explain the majority of diseases that we find in society as a whole, especially if we're talking about where the majority of infectious diseases occur, which is in third world regions. So the biggest problem, at least in those regions, are mixed infections. In fact, there was a study recently done within the last 10 years. There's this long longitudinal study really investigating just thousands and thousands of children in all of these different regions around the world, finding that especially in third world countries, when these children succumb to diarrheal disease, which is really, really frequent, most kids experience diarrheal disease within the first year of life multiple times in third world regions. And they find that in these kids, when they experience this diarrheal disease or gastroenteritis, it's not almost ever caused by one particular pathogen, like we just talked about. You could find Salmonella or you can find E. coli, but you can find it with 2 or 3 or 4 other pathogens causing the same kinds of disease. This is called a mixed infection. So it's very, very challenging to treat these types of infectious diseases, right? Because if you kill or eliminate one pathogen with an antibiotic, for example, it doesn't do anything to the actual disease because the person is still infected with 3 or 4 other pathogens causing the same disease. Other than that, in our region, in more developed cultures around the world or countries around the world, we oftentimes experience something called microbial dysbiosis, which we're going to talk about next, which oftentimes leads to low levels of gut inflammation, which causes increased susceptibility to pathogenic infections, but also has significant detrimental consequences to whole body systems. So this really, you cannot identify one pathogen that's leading to this condition. A really, really classic example of this that's a very modern disease that is really afflicting like tens of thousands of children in the developed region is necrotizing enterocolitis. This is a disease that afflicts preterm infants primarily, very, very rare for full-term infants, but for preterm infants, especially those born earlier than 33 weeks of gestational age, about 20% of them, sometimes the numbers are much higher, sometimes lower, depending upon the hospital, but about 20% of these infants will succumb to necrotizing enterocolitis, which is a huge damaging disease of the gastrointestinal system, specifically of the small and large intestine, causes major bloating and a huge influx of immune components and usually kills the infants very, very quickly within 24 to 48 hours. And what they're finding is exactly this, that it's caused by kind of general microbial dysbiosis, which is a shift in the balance of the good guys and the not so good guys, and gut inflammation. That very rarely is there an actual pathogen that's isolated from this region of the body. So this is really why I focus on this so much in this course, is because so many of the infectious diseases that we're seeing today, or many of the diseases, inflammatory-based diseases that we see in our society today, are not caused by one pathogen. And really, it's based on this concept right here, dysbiosis, a shift in the balance of the good guys, which we call the symbionts, the ones that we just talked about, which I called the normal flora, and something called the pathobionts. A pathobiont is an organism that really does not produce any benefit, but it can lead to inflammation and can alter metabolic activity and can increase in pathogen and colonization. So in this picture here, it's a good image. This is from a Nature immunology review, where in most people, like you and I, we exist in this state of equilibrium or symbiosis, where we have a significant number of these guys that we just finished talking about, all the normal flora that bring regulation, right? They bring benefits, they improve metabolism, et cetera, et cetera. There potentially is a subset, and this is arguable here. In fact, I argue that this doesn't exist. Potentially there's a subset of these guys that exist in the body and bring neither harm nor benefit, right? They kind of sit in the middle, and that's the term commensal bacteria. And then over here in this arm is the pathobionts. These ones are things like actually E. coli that I mentioned. So family or members of the family Enterobacteriaceae. Enteric bacteria, they're gram-negative, they have that lipopolysaccharide layer, and they usually are kept in balance, but sometimes if that balance is shifted, this is with perturbations like you take antibiotics, you eat a diet that's very poor, very low in fibers, very high in simple sugars, then you're really promoting the growth of these types of bacteria. In and of themselves, they're not terrible for you, but they can really increase the inflammation in the gut. And inflammation is one of the leading causes of tons of different disorders, like cardiovascular disorders, brain, cognitive developmental disorders, immunological disorders, and really, really opens the door for pathogenic infections. So pathobionts are not pathogens, but they can open the door for pathogens through this increase in inflammation. So what, again, causes this dysbiosis? Well, over here on this side, on this arm, right? An unbalanced diet is one of the biggest factors here. Right? Again, if you're eating a diet that has low diversity, if you're eating a diet that has simple sugars, right? Basically the Western diet. So high in carbohydrates, high in animal fats, very low in fermentable fibers. This is going to really push your normal flora to those pathobionts, really, really favor that type of diet. And your beneficial bacteria don't do so well with that particular type of diet. And so if you have that, then you're going to have dysbiosis, which can lead to a number of these different things. Cancer even is on this list, inflammatory bowel disease, irritable bowel syndrome, allergies, diarrhea, and even pathogenic infections here. Severe stress can cause this shift. Aging in and of itself does really push the shift of that balanced microbiota. So this is something that has to be combated with a significant input of beneficial dietary components. Antibiotics, I've mentioned already, is a big perturbation, so that means just like a disruption in the microbiota. And then even infections. So if you do have an infection, even upper respiratory tract infection can cause a little bit of a shift in your flora, in your gut. So all of these things need to be considered. You can recover it through changes of these things, interventions like probiotics or prebiotics. And if they're not changed, right, then these are some of the consequences of this dysbiotic community. And then one more thought on this human microbiome. So this microbiome, when we're talking about this, technically a biome as a reference to the number of genes or DNA, right, a genome in the microbial community, whereas the microbiota are the number of living organisms, different types of species we have. So just to consider this, the microbes inhabiting you contribute their genomes to yours. For every human gene, so for every one of our genes in our genome, there are 360 microbial genes, right? Us and them. And these microbial genes, right, not all of them contribute to human function, but a great vast majority of them have direct impacts on human health and metabolism. So just keep that in mind. What you are feeding your gut bacteria and providing as opportunities for other organisms in your body have direct consequences on your genetic expression as a metagenomic organism. So finally, we are at the hygiene hypothesis. And what is this concept? It is the idea that early exposure to microbes stimulates the immune system, but that overly sterile environment leads to allergies, asthma, and the list goes on. Right? And this concept has been altered somewhat. Now, it still is a hypothesis that exists in scientific community, and it is investigated some. We do know, as mentioned already, that normal flora are vital for immune development, right? So the beneficial flora that we've discussed already, the fermenting, the organisms that can ferment fibers. So by putting dietary fibers in your diet, you can promote the growth of these beneficial bacteria. Even as an infant, right, the dietary fibers exist in human milk. And if a baby is given formula without these dietary fibers, they really do have some severe consequences on immune system development because they don't have the same beneficial flora to promote that immune regulation. So there is a significant difference in the infants with different dietary input. And this early exposure is really, really important to developing tolerance, but it's not just in that first window of life, not just in that first few weeks. This really is that first few couple of years of the infant's life. In fact, what we found is that for an infant, their gut bacteria really shifts back and forth with all kinds of different inputs, right? With environmental exposure from caregivers, etc. Dietary components definitely have a huge impact. Any kind of infection that they face, and all of these different factors until they're about the age of 2.5 to 3, wherein the microbiota, whatever they have at that age, seems to pretty much be what we have as an adult. So whatever your microbes look like when you were 3 is pretty much the same microbes that you have in your gut today, unless you have undergone a significant dietary alteration that has persisted for a long period of time. In that case, you can keep the changed microbes, but once you change your diet back to a standard diet, then it will revert back to what you had when you were 3. It's quite an interesting phenomenon, and scientists are still trying to investigate why that is. But all of it really has to do with immune system exposure too, and the development of your immune system. So it does make sense that if you prevent the infant from exposure to environmental organisms that are both like beneficial and potentially not so beneficial, that training, that immune system development early on in life really does not have the opportunity to just like test things out and see what's good and what's not so good and how it's supposed to respond to different things. And so there is an aberrant regulation, right? There's an aberrant response in those individuals that are not exposed to environmental organisms early on in life, so that by the time they reach that pivotal age where everything is sort of set in stone, not just the microbiota, but really how the immune system is going to function for the rest of your life. If they haven't had that early training, then they're going to have a significant consequence to their immune regulation. So it's an interesting idea, and there are kind of spin-offs of this theory. There's one called the missing microbes theory, or something called old friends hypothesis. Both of these things kind of spin off the same idea here, but a little bit different. In that regard, it's more along the lines of we have essentially over-sterilized, used too many antibiotics. We've really wiped out some of the beneficial bacteria or even parasites that existed in our community and were really important in that immune regulation early on in life. And without those microbes, the missing microbes, then we have really a disadvantage in the early stage developmental period, which is one of the foundational reasons why we have a significant amount of childhood disorders that were never seen prior to the advent of antibiotics, especially, but really even prior to the kind of newer age of over-sterilization. And this last slide is just an interesting discussion. You're welcome to download this video and watch this yourself, but this is really just an opportunity for you to think about this idea of over-sterilization and perhaps the hype of marketing that's gotten us to where we are today.